Welcome to this cryo-EM course. Last class I discussed about the entire anatomy of the electron microscope, where we connected all this how it can connect with the gun deflector, beam deflector, and how objective lens is connected to focus, C2 lens is connected with your intensity, and condenser lens is connected with your C1. But now previously also I discussed about that how one electromagnetic lens can converge and diverge the beam, and also sometime we are talking about the weak lens system. Strong magnetic field or strong lens is how, when we are using a strong lens or when we are using a weak lens, what kind of image formation will be there. In this particular case, we will learn one more term, that is called the defocus. In the electron microscope, when we are doing cryo-electron microscopy, most of the time we are talking about the defocus the beam. So, today we will learn that, but before that, if we want to discuss about that image formation, first we have to know the ray diagram, how we can do a ray diagram and how this ray diagram is helping us to get the images. So, let us do these things. So, I will go little bit slow because the first time we are doing this ray diagram and many things is depending on this ray diagram. So, it will be better to understand those ray diagram carefully. So, first thing first, we will just draw about some kind of image, that when we are imaging something in light microscope, it will be a similar kind of imaging system we are going to use it. So, I will just draw the optical axis first. So, just consider that okay, this is the straight line and this is my optical axis. Okay. And now I have an object here, and then we have an electromagnetic lens. This is my electromagnetic lens. This is the axis of the electromagnetic lens. Just for a minute, this lens is not looking very symmetrical lens. So, this is my magnetic lens. Okay. So, when we have this magnetic lens, we all know that electron beam, it will incoming electron beam, if this is the object, from one point one electron beam will come this way and then they will converge. If this is my plane, I will just consider this is my image plane for now. Okay. Now, there is an incoming electron beam, and that electron beam can travel any path. Okay. So, and then it will go through this particular point and it will give us an image here. Now, we are getting an image somewhere, but if this is the optical axis, if we just consider that okay, so this is the ray, it can come, it can merge here, but there will be multiple here. We want to see how it is forming an actual image, right. So, we will just follow the normal protocol, how it is going for the normal light optical microscopy system. So, one ray, it will go through this axis and it will come here. So, in this particular case, these rays will come here and merge and give us an image here. Now, in this particular magnetic field, when we are using this magnetic field, there will be a crossover point here, and this is, we can say, the focus point in this particular magnetic field. Another possibility is that okay, if I have a little bit stronger magnetic field, stronger magnetic field means it is pushing this electron beam towards this direction, and you can see there is a merge and it will give us an image somewhere here. This is the first focal length, if we just consider, and I will say this is f1. Now, this blue if we just consider, I can changing this focal point again, and this is my F2. Now, if I just use the more stronger electron beam, what will happen? This will go and merge somewhere here, and then definitely it will give us an image here. So, you can see that in this particular case, this is my F3. So, now you can see that at the beginning when we are starting this one, this is our image. We can consider in the second one that when we are using strong electron beam, this is my second image. When I am using more stronger electron beam, this is my third image. In all the cases, this is my image plane. If I just consider, this is my image plane. Okay, this is my image plane 1, this is my image plane 2, and this is my image plane 3. Okay. So, now this is my object. So, what you can see here, the most important part, that okay, there is a focus point or focal point that is changing, and that is basically changing based on the magnetic field. If we have a very weak magnetic field, converging point is somewhere here. If we have a very strong magnetic field, the converging point, it is going up and it is giving us an image here. So, that strong magnetic field, our image size is smaller here, okay, when we are using only one lens system, and then when we are using a weak lens system, we can see the image size is increasing. Now, if we just consider that okay, this is my D0. So, D0 is the distance between your object and the lens system, and then the first one, this is my imaging plane, the first image. So, that I can tell this is distance from the lens to image, that is Di, and then this is my first image. Then this is my second image. So, I can say Di2. This is my third image. If I just consider, so this is Di3. So, you can see this distance is changing. Now, from this one, if I want to calculate the magnification, I can tell magnification, it will be like that, first Di anything by D0, that will give us the magnification, exactly same like our optical microscope. Now, if we want to calculate the focal length, we can say that D0 plus 1 by Di. This is how we should calculate this one. So, this is our first page. Now, we will go to the next one, that we want to draw another complicated system in electron microscope, how the image should look like. Then finally, we will come and draw a aperture, and we want to see what kind of differences are there when it is forming an image, or how these electron beams are coming. Okay. So, if you remember last class when I was discussing that okay, this is an optical microscope, and if this is an object, we are getting an image, it is going through the center and it is giving us an image here, right. Now, it is only 2 days we are using, or maybe you can consider 3 days to describe these things, because it will be a clean thing. But actually, when we are trying to do these things, basically from this particular point, rays are coming in all directions, and then it is giving us an image here. All the rays are converging in one particular point, and then it is giving us an image. Now, if we just consider that, okay, so here if we just this particular point is only one rays is coming, no, here also there is a multiple rays are coming and all are converged in this particular point, which is giving us this final image. What about the middle point then? So some kind of center point if we just consider, or if we just consider the center point here, there is a different color I am just trying to use. So rays will again come at different orientation, right. Now all are converged in one particular point and give us this. So whenever there is a object, rays can come out from any direction, and that will converge in this and give us this particular image. Now this is true for electron microscope sample also, but if we draw the ray diagram like this, it will be very difficult for anyone to understand what is going on there. So definitely this is not the best way to draw, always this is the best way to draw it. So let us go back to normal electron microscope and explain how we can use couple of rays and draw a ray diagram and get that image. So here first let us draw the optical axis. Now let us draw an object. So something this is our object. So rays can come from any side. Okay. So in this particular case our lens system is somewhere here. Okay. So first let us draw that axis which will help us to draw the diagram. Now I will use one particular blue color here. What will happen? There will be a one possibility that one straight line it will come here. Okay. The same thing is true for the other part, then one straight ray can come here. Now, so in one particular part we are just drawing using all blue colors. So here there is a possibility that ray can come in this direction also, right. It can come all the directions. So that same thing is true for the red ray also, that whatever rays are coming from one particular point here, it can go all the way here and all the way here. Okay. Now when it is traveling and it is going to give us an image at the bottom at the image plane. So if we just consider the straight line, first let us consider that straight line, and let us consider that this is our imaging plane. We want to get that image. So there is a, as we mentioned, that rays are, so this is our object, you can consider that this is one circular part and here there is a one square. So we are expecting that this square it will come here and this circle it will come here. So let us first draw this blue rays and see it, because this is a straight line, it will be very easy to bring it down. We will just draw this straight line. We are expecting that image is going to form here. What about that red rays? If we just consider this red one, it will also travel in same way and merge this particular point, right. So I will just bring it down little bit, so it will touch the image plane. Now we have other rays also, we have to satisfy those one that should come here and merge one particular point. So, all the rays it will come and merge here. So, this will merge somewhere here, and there is a other ray it is there that can also go all the way down and merge somewhere here. Then what about the red ray? Now here that is deflected in a certain position. So, it will come like that. So, it will come here and then at the end it will merge, right. What about this ray here? This ray also come and merge in this particular point. Now, we are reaching a very important point. So, in the red point it is coming. So, this is my sample. So, electron beams before that all are coming like this, and we are considering these are the parallel electron beam before interacting with sample. Okay. So, always remember this, we are always considering our electron beams are parallel electron beams. What these electron beams are coming from? These electron beams are coming from C2 lens. C2 lens means intensity. So, first it is going to C1 lens and then it is going to the C2 lens. So, we always discussed the electron beam, it will be converge and diverge. So, it will converge somewhere here and then again it should diverge. But there is a system with C2 lens or for 300 kb C3 lens or for 200 kb C2 lens and upper objective lens. This C2 and upper objective lens together, we can use it in a such a way that we can make this electron beam parallel. So, it will converge, then when it will come near the C2 and upper objective lens, these two it will make the electron beam some sort of parallel, and this parallel electron beam it will go through the sample. So, we will discuss these things in next class, not today's class, how we can get the parallel electron beam. Only remember that parallel electron beams are very important when we are doing the imaging, because we do not expect any kind of deviation or scattering of the electron beam before it is interacting with the sample. We are considering all the electron beams are very coherent and it is coming as a parallel electron beam before it is interacting with the sample. When it will interact with the sample, then there will be a deviation, and that may be a unscattered electron or scattered electron, elastically scattered, non-elastically scattered. We will bring all of these things in our account to calculate the image. But before that, beams will be always parallel. Now, if you look at here, this is our sample. I told the beams are parallel. Now, this is the electromagnetic lens system. Now, in this electromagnetic lens system, you can see one very important point when these rays are going, that is converging in one particular point. So, here you can see all these things are coming in one particular plane. So, this is forming one dot, this is forming one dot. This is the plane, it is called back focal plane. So, this back focal plane, it is called we are recording the diffraction pattern. So, diffraction pattern we are recording in the back focal plane. How? I am coming to that. Now, this is called the imaging plane. This is called the imaging plane where we are getting the image. So, if we have a object here, you can see this is the square, this is the circle, and this is the object, and object is much bigger than image is much bigger. So, this is our image. Now, if you just calculate this distance, this will be d0, we already discussed about that, and now this will be our di. So, if we want to calculate the magnification, this will be di by d0, and the focus of this lens, this is called the focus of that lens. Now, this is an important plane, what is called the back focal plane. Back focal plane, we are collecting all the diffraction pattern here. How? If you look at in this particular point, so this is one of the first imaging plane of our microscope. After your gun lens, condenser lens 1, condenser lens 2, then your objective lens, and this is our first imaging plane where we can record the image. It is the first place where we are getting some information about our object, and here we can record the diffraction pattern. If you look at, so when we have a image, what is happening, all these 3 rays are coming one particular point, and at the end, because of this lens system, it is converging in one particular point here. And similar thing is happening, this is a point where we are expecting a square. If we look at this, this is a square and this is a circle. So, all these rays are coming from the circle, merge here and giving us circle. All these rays are coming out from these squares. So, all these are blue color, this is a blue color, from the square it is coming, and these blue colored rays are coming and converge in one particular point and giving us this square. Now, if we have this star here, so rays will come from the star, and the star it will appear here. So, in the same way, all the 3 rays it will come. So, one rays it will come here, one rays it will come here, and other rays it will come in this direction. So, if we just draw this one, all the rays it will come like this, okay, and they will go like that and converge in this particular point, right, and then it will give us this star at the center. So, this blue square, it is giving us a blue in this particular point. This circle, it is giving us a circle somewhere here, and this pink star is giving us a pink star here. Now, what information we are getting here? We are getting this information. You can see that one ray it is coming from that circle, one ray it is coming from a star, one ray it is coming from this blue square. So, this is a mixture of information from this blue box, star and the circle. Same information we are getting here. What we are getting? We are getting some information from these blue rays, some information we are getting from these red rays, and some information we are getting from this pink. So, this is a point, it is a mixture where some kind of scattered electron beam are coming from the square, star and red. Same point is happening here. So, what we are getting? We are getting some kind of bright spots there, and its intensity may be different, but it will be some kind of bright spot all over based on my sample like this, and that is called the diffraction pattern of our sample. Most of the time biological samples are amorphous sample, and that is the reason their scattering is very poor, and you will not get a very strong diffraction pattern. But whatever we are going to record here, we are going to record the diffraction pattern, which is basically information from multiple points. All this information are coming together and merging in one particular point. So, you will get a sometime a very bright spot, sometime a very light spot at different position. And when you are recording this kind of diffraction pattern, if you just call this is a diffraction pattern, what I told, sometime you will get a very bright spot at the center, because all the three rays are coming together. If you have multiple rays here, now if you just consider, okay, some rays are coming here, some rays are coming here, all are going to merge here. So center point you will get a very bright spot, and then you will get a different kind of intensity and different kind of spot. So those are the electron diffraction from your sample, and this is the center area, you will get a very bright spot, and the rest of the part it will be less intense. And if you look at this, sometime we can tell that this is a power spectrum, that we are going to discuss in the later stage. What is power spectrum? That is coming from the diffraction pattern and the intensity of the diffracted spot. Here if you look at this center spot, if you remember that I was drawing something like this kind of curve, you can see center always there is a very intense beam. So if I try to draw a curve here, we will always see this will be the maximum and then gradually it will go down. So you will get it like that, you will get a maximum peak here and then gradually it will go down. Why it is gradually going down? We will discuss these things in the later stage when we will talk about the data processing. But if you look at in this diffraction pattern, you can see the center we have a very bright spot. This is called the low frequency signal, and this is called the high frequency signal. Okay, and these high frequency signals are very important to get the high resolution information of the sample that we are going to discuss in the later stage. Okay, let's discuss little bit about more about our focus and defocus, how we are considering the focusing the beam. Before that, let's discuss little bit more about when we are using an aperture, what kind of how we are going to block the electron beam. Okay, so far we discuss about the specimen, different kind of diagram, how the samples are here. This is an image, object. This is my object. Okay. Now this is my optical axis and it is going to give us an image. So previously I showed you, when you have a object and we are expecting electron beam from all the angle and they are going to merge in one particular point and they are going to give us an image, right? And this is our diffraction pattern. Here we have a lens system. So this drawing is not that perfect because by hand I draw it. Now if this is an object and you are getting an image here, if you do not have an object, what do you have? If you just consider, if you just remember the filament, first electron rays are coming and merging in one particular point, and that is we are talking about the D0, which is regulating our intensity. Now this D0, if you just consider, this is just a spot. So if you do not have a sample, if you are just trying to image a spot, you will expect a spot after converge, diverge, whatever it is happening, you will get a spot. So here we are not using any this kind of sample, we are just considering, okay, this is the specimen is everywhere and our beams are comes out from this particular point and how it converts to a image plane. Okay. So if this is our sample, so here this is our optical axis, this is our optical axis, and let's consider that this is our specimen and the electron beams are coming out from this particular point. It will go in a same way, in a very symmetric way, and then electron beam comes like that and converge in one particular point. Now, so when the electron beams are coming, it can come this way also. No one can stop that electron beam. So, electron beam can come in this way also, and then they can converge in this image plane. So, in this particular case, you can see that beam is deflected too much. If I want to converge this particular beam in one particular point, if this is the image plane and I want to converge it in one particular point, for this blue rays I need very strong electric field, magnetic field. Magnetic field is required to converge. So, comparatively this red ray, it is close to the optical axis, we do not need that much, it is not deviated that much. Now, let me draw the one more one. So, if we have another ray, this is another ray, it is coming, another ray it is coming, and it converge in this particular point. So, when rays are near the optical axis, deviation it will be less, we do not need that much magnetic field, but when the rays are deviated too much, sometime we need a stronger magnetic field. Lot of time, if there is a scattering is too strong and it is scattered too much and that much magnetic field we do not want to use it, or those rays if it is not that much useful, if you just consider that this blue line or partially this red is also not very much required for us, if this is very strongly scattered, we can block those kind of electron beam. So, what we can do in this particular case, we can use some kind of aperture. So, this aperture it will come like this and it will block some kind of electron beam. So, here you can see whatever electron is coming in this particular region, all are blocked by this aperture. So, it is only allowing those electron beams, if I just consider that through this aperture only these blue electrons can go through. This blue So, in this particular case only that blue electron can go through. Here also you can see there is a blue rays which can come here, which can come in this particular point, which can come in this particular point and give us an image. So, using that aperture we are blocking lots of unwanted electron beam, and sometimes this is helping us to increase the contrast. This is the function of the aperture, and this will be really helpful for biological samples. So, when we want to image, here we already discussed about the C1 is a spot size, right. So, the spot size, and then C2 is intensity, and then this aperture. If we just use this kind of aperture, we can block lots of electron dose, and then we can control the electron dose. So, I will stop here. In the next class we will discuss about the focus, defocus and over focused and under focused images using the ray diagram. Thank you all for your attention.